Hemostasis is a process that's carried out by our platelets and some of the soluble fibers that can be found dissolved in the blood plasma. Hemostasis can be defined as a set of actions carried out by our body in the interest of stopping excessive bleeding. The whole purpose of hemostasis is to prevent a hemorrhage, which is the loss of very large amounts of blood from the blood vessels. In order for this process to be effective, hemostasis must be quick, localized to the specific region that's damaged, and carefully controlled, as not to cause a systemic clotting of all of the blood in the circulation. The three mechanisms that make up hemostasis as a whole are referred to as our vascular spasm, platelet plug formation, and the blood clotting or coagulation cascade. In sequence, these three processes help prevent large loss of blood from the blood vessels. We will discuss each of these processes now. The first mechanism of hemostasis is the vascular spasm. This refers to a contraction of the circularly arranged smooth muscles that are found lining our blood vessels. As the muscles around the vessels start to constrict, that's going to close off any potential damaged spots in our blood vessels. As there is a contraction or a vasoconstriction of the muscles in our vessels, that's going to narrow the vessel and therefore make it less likely for blood to exit from the vasculature. This process can occur from several minutes up to several hours. The second step of hemostasis is referred to as the platelet plug formation. The platelet plug is a building up of platelets in the blood. Different reactions are going to occur that will make our platelets very sticky, and they're going to bunch up and form this big plug to prevent excessive loss of blood from the vessels. The way this process occurs is that when the endothelial cells of the blood vessels are broken or damaged in some way, they will expose the collagen fibers that are inside of those cells. Platelets can react and stick to the collagen fibers, and this process is referred to as platelet adhesion. During this process of platelet adhesion, the platelets are going to start to extend many different projections that will increase contact with other platelets and more collagen fibers. They will then start to release certain chemicals that will sustain the vascular spasm. Specifically, the grouping up and sticking of the platelets together can be referred to as platelet aggregation. When the aggregation gets big enough, we then will refer to it as the platelet plug. Especially in small blood vessels, the platelet plug is a very effective means for preventing excessive blood loss. You can see this process listed on this slide. First, we're going to have platelet adhesion, where platelets will actually make their way to the damaged endothelial cells and react with the collagen fibers that can be found exposed from those damaged endothelial cells. Eventually, as these platelets are sticking together, they're going to start to extend little processes out of their membranes, and that's going to increase contact between platelets. So we have more surface area for contact with the platelets in this manner. The platelets will then start undergoing what's known as the platelet release reaction, where they start to release different chemicals into the bloodstream. ADP, serotonin, and thromboxane A2 are three examples of chemicals that are released during the platelet release reaction. It's probably a good idea for me to go ahead and highlight the three of those: ADP, serotonin, and thromboxane A2. These chemicals are going to activate more platelets and sustain the vascular spasm, until eventually we get platelet aggregation, where we have many platelets that are all bunched up and they're blocking the hole that has formed in the blood vessel. This is our second step in hemostasis. Our third step is what's referred to as coagulation, or we can call it the blood clotting cascade. Now, there are actually two different pathways that will lead to coagulation: an extrinsic pathway and an intrinsic pathway. However, both of those pathways will end up converging and forming the common pathway, which is what is shown on this slide. This is a cascade that starts with a molecule known as factor X. And once factor X is activated, it is then going to begin this common pathway cascade. Factor X, once activated, is able to eventually go and activate the prothrombin activator. That's called prothrombinase. I've gone ahead and written this so that you can see this, but you'll also find this reaction shown on page 685 in your textbook. So once prothrombinase is active, which is the prothrombin activator, prothrombinase is then eventually going to convert another molecule known as prothrombin into its activated form known as thrombin. When we see pro in front of a molecule's name, that means that it's a precursor, immature form that's inactive. So prothrombin will eventually get activated and become thrombin. Thrombin, in return, will eventually go and convert another set of chemicals. It's going to convert fibrinogen into another molecule known as fibrin. And fibrin is the major player for coagulation. Fibrin is the soluble fiber that will eventually form the blood clot that will ultimately stop the bleeding. So this slide just shows this in a different picture. You can see that there's an extrinsic pathway and an intrinsic pathway. All of this stuff up here is not what I'm really going to stress on your upcoming exam. But I do want you to see that there is eventually going to be an activated factor 10. Or we can also call this factor 10A for factor 10 that's activated. Factor 10A is going to activate the prothrombin activator known as prothrombinase. Prothrombinase, when mixed with calcium, is going to convert prothrombin into thrombin. And then thrombin is eventually going to go and convert fibrinogen into fibrin. And fibrin is what's ultimately going to lead to the blood clotting. You can see from these three pictures that in the early stage, there's less fibrin, but eventually we start to form more fibrin. And we form this meshwork, and it allows for our red blood cells and other things in the blood to get caught up and aid in the formation of the blood clot. Oftentimes throughout the day, occurring most frequently at sites of minor roughness or perhaps areas where plaques are beginning to form inside of the blood vessels, tiny blood clots will start to form. And as blood clotting is a positive feedback mechanism, and the actual coagulation is amplified the more clotting factors are involved, clots normally have a tendency to grow in size, and that can eventually lead to issues of clogging even undamaged blood vessels. So that's where the process of fibrinolysis is going to come in to prevent this unnecessary blood clotting. Fibrinolysis can be defined as the dissolution of a blood clot. The way this process works is in clots, there is going to be a plasma enzyme known as plasminogen that's incorporated into the blood clot that was forming. Plasminogen can eventually be activated into what's known as plasmin. And plasmin is an enzyme that will eventually start to break down these undesirable clots. So plasmin is a very important regulator to prevent excessive blood clotting, which could potentially lead to other more dangerous conditions. Now we have two factors that I really want to mention, procoagulants and anticoagulants. And you can probably get a gist of what each of these groups of factors are going to do. The procoagulants are going to increase and enhance the blood clotting activity, and anticoagulants are going to prevent and inhibit blood clotting activity. Procoagulants are used when we're trying to speed up the process of coagulation. Vitamin K is one, for example, that will directly lead to an increase in clotting factors. Vitamin K is necessary to synthesize 4 of the clotting factors that play a role in coagulation. Vitamin K is normally found within the body, and it's considered a natural procoagulant. But two artificial examples of procoagulants are two things that we often find ourselves doing when we are trying to prevent bleeding on our own or perhaps on somebody else. So I find it very likely that you've perhaps utilized either of these techniques. So a gauze is often used when patients are bleeding. And this is not to just prevent a mess from getting on the floor or preventing a spill in the room that you're in, but a gauze is actually used to give the platelets more of a surface area to bind to. It gives the platelets an area that they can stick, and this will help to keep more platelets in that particular area and speed up coagulation. Pressure is another factor that can be considered a procoagulant. Pressure on wounds is actually going to mechanically disturb that area, and that releases a molecule known as tissue factor, which will eventually aid in coagulation as well. Now these are some examples of procoagulants, but we also have examples of anticoagulants, which will do just the opposite. These are going to prevent blood clotting. As I mentioned earlier, blood clotting needs to be a controlled process, and we don't want this to happen unless we have some type of damage that's occurred to one of our blood vessels. So the endothelium normally has characteristics that push platelets away. The endothelium and the platelets don't normally like to stick together. But eventually, once those endothelial cells are damaged, they're going to expose other factors that will facilitate this blood clotting cascade. Another example of an anticoagulant is vitamin E. Now, these are two examples of natural anticoagulants, but we also have certain medications that can be classified as anticoagulants as well. And one of them is known as heparin. Now, heparin is normally produced by several cells in the body, but we can also administer heparin to prevent blood clots as well. So blood clotting is a process that requires very specific regulation and exceptional precision. However, as with most things in the human body, all processes can go wrong to some degree. So if blood clotting is a little out of control, we have a couple conditions that we see that will relate to this. So the thrombus and the embolus are two examples of undesirable clotting instances. Both of these are very similar, but a thrombus refers to a clot that forms in an undamaged vessel, and this eventually, if allowed to persist, can form a blockage in that vessel and prevent blood flow to certain areas. Similarly, if a thrombus forms and we have this large blood vessel, pieces of that thrombus can break off, and we then start to call that an embolus. If little pieces of that blood clot have broken off, those blood clots will travel throughout the bloodstream and perhaps get lodged in other vessels. And if that clot gets lodged in a vessel that can be found in the brain, that can lead to a stroke. Areas where blood vessels are especially narrow are causes for concern for an embolus. So one of the treatments that we will use to prevent this are going to be our anticoagulants, such as heparin, another one known as warfarin, and aspirin. So if blood clotting is exaggerated, we may end up with a thrombus or an embolus. However, there are other conditions that refer to an inability for us to properly facilitate the coagulation cascade. We have thrombocytopenia and we have hemophilia. Thrombocytopenia refers to a decreased number of platelets. And naturally, as you can assume, if we do not have enough platelets, we're not going to adequately be able to facilitate the coagulation cascade, as platelets are a major player in carrying out hemostasis. Similarly, hemophilia is also a condition that causes a prevention in blood clotting. Hemophilia is a genetic condition where there are no clotting factors present in the blood. So with hemophilia, if blood vessels are damaged, the patient ends up continuously bleeding and unable to clot blood at all. One of the last major concepts that we're going to cover here in Chapter 19 are the blood groups and blood types. The understanding that we have of different blood types gives us the efficacy to be able to transfuse certain patients' blood into others, and this, in many instances, saves lives. The theory behind blood typing stems from very special proteins found on the cell surface of our red blood cells, known as antigens. Antigens are little glycoproteins or glycolipids that stem from the cell surface of our erythrocytes. Antigens can be found on all different types of cells, including virus particles as well as bacterial cells. And antigens are also found on our own cells. These can be used as cell identity markers. But we'll also find antigens playing a very, very major role when it comes to our body's immune system. We'll find that antibodies, a very specialized protein that is functioning for our immune system to help us find and detect certain foreign invaders, will be stimulated by antigens. Now the next slide will really go into what antigens can be found in the ABO system. However, based on a few antigens that we have, we get an entire blood typing system that is considered the normal convention of today. This is based on a few antigens, namely the A antigen and the B antigen. Each of these antigens has a different molecular structure and therefore is going to activate different types of antibodies. It's very possible that we can have a mixture of both, and we would call those individuals AB. And we can also have neither, and we refer to those individuals as O. So the A antigen and the B antigen are the major antigens that are going to be important for our blood typing concepts. There is a third antigen known as the Rh antigen, and whenever the Rh antigen is present, we say that that blood type is positive, and when the Rh antigen is not present, we say that that blood type is negative. Now, the Rh antigen is found in most individuals. However, antibodies to this particular antigen are not normally found until our bodies are exposed to the Rh antigen. And that will be discussed a little bit later. But what we see on this slide is a representation of what types of antigens we can find on our red blood cells and how we're going to understand how blood typing works. So you can see this red blood cell has a different shaped antigen. We can see this is the A antigen. It has this little ball at the end. And the B antigen is left with a little arrow or a triangle at the end. And these are two different proteins with different molecular structures. You can see that some red blood cells can contain both, and others can contain neither. And each of these blood types is listed above. If you have the A antigen, you would be considered type A. If you have the B antigen, your blood type would be type B. If you have both, you're AB, and if you have neither, you're considered O. Now, what's also important for us to understand are the antibodies that are associated with antigens. Antibodies are normally formed by our immune system to things that are not considered us. We would refer to those things as non-self. We make antibodies to things that are different from our own body structures so that we can identify foreign things when they make their way into the body. It would not be very beneficial for us to make antibodies to the own antigens on our own blood cells, as they would eventually react and cause an immune reaction. So what we're going to find is that the antibodies found in the bloodstream of individuals that are type A will only be the B antibody, or the anti-B antibody, if you'd like to say it that way also. This ensures that the A antigen is not going to be attacked in the bloodstream of this first individual. However, our body will be protected against foreign antigens, and that would be the B antigen. Naturally, the B antibody would bind to the B antigen and cause that immune reaction. And that's why we see the anti-B antibody that's existing in the bloodstream of the type A individuals, and we see the anti-A antibody in the bloodstream of individuals who are type B. Now, when we look at the individuals that are type AB, they will have both the A and the B antigens on the surface of their red blood cells. Therefore, since we have both of these antigens, we will have neither of the antibodies, as that would cause an immune reaction if we had either of those antibodies. And you can see individuals that are type O and have neither the A antigen or the B antigen will contain both the A antibodies and the B antibodies that's floating around in their plasma. So the convention is, if you have the antigen, that antigen would indicate what type of blood you are. So if we have a sample of blood from our patient, we can actually perform a series of tests to determine what blood type that patient is. So I've just added each of these letters in here, as these were unfortunately cut out from your book. On page 690, you will find this picture in your text with each of these labeled. I'm not sure exactly why they got cut out, but I've just taken the liberty of adding them in so that you can see each of these respective blood types. So the way that we will assess the specific blood type of a patient is by adding a substance known as antiserum to the blood, and we're going to look for what's known as agglutination of that blood, and agglutination will indicate a positive result for that antigen. So let's make a little bit more sense of this. We start with a sample of blood. We'll see that we'll take this individual. This is going to show us what a blood stain would look like from a patient that has A blood. So we take a drop or a couple drops of the A individual's blood, and we drop it here, and we drop it here, and we drop it here. Now I'm telling you that this individual is A, but before we've actually figured this out, this is the series of steps that we're going to do. Before we know that this is an A individual, we're going to take a few drops of their blood, and we're adding this antisera to determine what the blood type is. So the antiserum contains specific antibodies. You can see this is an anti-A serum, and this is an anti-B serum. Anti-A serum contains anti-A antibodies. So therefore, if we have individual spots of blood, we can add the anti-serum and specifically test for that antigen on the surface of the blood. So this is what A blood would look like untreated. And in fact, most of these blood types are going to look fairly the same to the naked eye. But once we add the A anti-serum here, we're going to look for what's known as agglutination. Agglutination is a process that's carried out by antibodies where antibodies will bind to antigens and clump up. And they'll form this mass of antibodies and antigens together. Since this is showing us untreated blood, this is what it will look like normally. But you can see some positive results for agglutination in this well right here. Here with the B antiserum, we will see no results, which indicates that there was no agglutination. So what's truly going on here is we have this sample of blood, and we know that since I wrote this in here, that this is an individual with A blood type. So when we add the antibodies, the A antiserum containing the A antibodies, that's going to react with the A antigens on the surface of these red blood cells. Now, since the B antibody did not react with anything, that indicates that this blood does not normally have the B antigen. So since this blood type only has agglutination in the A well, that indicates that we're looking at a type A individual. Now for type B, you can see that we'll only have agglutination in the well that was treated with the anti-B serum. We have no agglutination here in the A well because there's not naturally any A antigens on B type blood. So that's why we'll see agglutination in the B well only. With AB, we'll see agglutination in both the A well and the B well. And with O blood types, we'll see no agglutination at all. Since O individuals do not have any antigens on the surface of their red blood cells, adding either the A antibodies or the B antibodies will not cause an agglutination reaction. And when you take the lab class, you will actually carry out this very similar experiment. So keep in mind, with the ABO blood types, if you have the antigen, you will not have the antibody. And if you do not have the antigen, you will have the antibody. You're going to have an A of something, but you will not have both. You'll either have the A antigen or the A antibody in your blood. Same goes for B, same goes for RH. So this is a nice slide to show you the characteristics of each blood type and what can be found in each. So a blood type A is going to have the A antigen, and it will also have the B antibody. It's not going to have the A antibody because that would react with the A antigen on the surface of those red blood cells. Remember, if we have both the antibody and the antigen, we get agglutination, and that blood is not going to provide us the transport and regulatory functions that normal blood is going to. The B-type blood is going to have the B antigen, and therefore will only have the anti-A antibody. AB blood types will have both of these antigens, and they won't have any antibodies because we don't want to have a reaction between either of these. O blood types will have neither the A or the B antigen, and they'll have both of the antibodies in their blood. Now, once we have determined what antibodies are floating around in the blood of a particular patient, we can use that information to determine which types of blood are compatible to infuse into that particular patient. So when infusing blood of one patient into blood of a second patient, we have to be cautious of the antibodies, and we want to prevent antibody-antigen reactions leading to agglutination in the patient that we are infusing. So we have to be aware that this A-type blood that has the anti-B antibody cannot receive any types of blood that have the B antigen. Therefore, our compatible donors are A types and O types. And using the same theory, we can see that the B blood type only contains the anti-A antibody, and therefore we can only use blood types of B and O. We don't want to add any A antigens since there is the A antibody found in that blood. A, B, and O are a little bit different. Since the AB blood type has both the A and the B antigens, and therefore will not have any antibodies, the AB blood type is considered the universal recipient. This means that the AB individual can receive all types of blood because there's naturally no antibodies for any of these antigens floating around in the blood naturally. However, the opposite is here for the O blood types. The O blood types are considered the universal donor. Because they're going to have both of these antibodies in their blood, they can only receive from other O individuals. So this limits individuals that are O blood type to only receiving from other O donors. So we have to be very careful with O individuals to make sure that we're not infusing any blood with these antigens on it that will naturally cause a reaction. So an easier way to look at this is the blood type A can't receive anything with B in it. So both B and AB will have the B antigen, and therefore neither of those are going to work. The opposite is true here. For the B blood type, anything with A is not going to work. The AB blood type is not going to have any incompatible donors because they will not have any of those antibodies in their blood, and therefore they can receive any types of blood that have any antigens on them. And blood types O are going to be incompatible with everything with the exception of other O individuals. So to finish up chapter 19, we're going to go over a couple clinical terms on the next few slides. One procedure we often see carried out that's related to blood is a bone marrow biopsy. This procedure is carried out by taking a needle and sticking it in and getting a sample of bone marrow. From the state of the bone marrow, we can use this to diagnose several conditions, such as leukemia, anemia, and others as well. A bone marrow biopsy is clearly going to be used to diagnose conditions that would be related to our blood cells. An exchange transfusion is basically what we were talking about on the last slide, where one patient donates blood to another patient. This can be necessary if one of our patients has lost a very large volume of blood and needs more blood fast. A hematoma is a fancy word for a bruise. Now, I don't want to underplay hematomas because some of these bruises can be very, very severe. So what we see with a hematoma is where blood has escaped the vasculature and accumulated in our body tissues in some place. Hemochromatosis is a specialized genetic condition where we're absorbing too much iron from the diet, and that is eventually going to cause a lot of other issues throughout the body. Plasmapheresis is a procedure where we take plasma, separate it from the blood cells, and then we return the plasma back to the patient. And then, once the plasma is separated from the cells in the blood, we put the cells back into the body. Septicemia, also known as sepsis or blood poisoning, refers to an infection of the blood. Now, a few slides ago, I mentioned that Rh antigen. Well, the Rh antigen acts very similarly as the A and B, however, it is a little bit different. Normally, even if we're Rh negative, we don't naturally have the Rh antibodies floating around in the bloodstream unless we are exposed to the Rh antigen. So this slide covers a condition known as erythroblastosis fetalis. This condition is focused on pregnancy. Normally, fetal blood enters into the mother's circulation. Now, it's very possible that a baby can be Rh positive while the mother might be Rh negative. So even though the mother is Rh negative, she wouldn't have normally had these Rh antibodies in her blood. However, once the baby's blood leaks into the maternal circulation, that exposes the mother and leads to the formation of the Rh antibodies. Now, typically, this is not as big of a problem during the first pregnancy as is the second pregnancy. Normally, the first baby is born without as many issues. However, during the next pregnancy, if that baby is also Rh positive, just as the first one was, we will now see that the mother has those Rh antibodies present in her bloodstream. This is an issue because the mother's antibodies are entering into the circulation of the fetus. So the Rh antibodies that have entered into the fetus end up attacking the fetus's red blood cells. So this can be very damaging to the second child. Specifically, erythroblastosis fetalis affects the second child specifically. There is a medication that we can use. We have a medication known as RhoGAM, and that is an antibody that's going to bind to and cover up those Rh antigens. and we can view that as allowing the baby to hide from the mother's immune system. Sickle cell anemia is another condition that we often see related to the blood. This is a genetic condition caused by a mutant type of hemoglobin called hemoglobin S. The result of sickle cell anemia is when red blood cells offload their oxygen to the tissues, they end up changing shape to this sickled form, hence the name sickle cell anemia. These blood cells last much less than the 120 days red blood cells are supposed to exist for. While sickle cell disease is a very serious condition, it can actually confer a bit of resistance to the parasite that causes malaria. I'll go ahead and write this to indicate that that is a very high yield statement. On that note, that will conclude our presentation on the blood. I will see you all in the next chapter, chapter 20, when we will discuss the heart.